Thank you for joining us today as we present, along with our friends from the Council for Federal Cannabis Regulation, on a very timely topic, quality standards and their regulatory impact on the cannabis industry. Before we begin, I wanted to cover a few housekeeping items. If you have any questions during the webcast, you can submit them through the Q&A widget at the bottom of your screen. Your questions will only be seen by the presenters, and thank you for those who sent in questions in advance when you registered. We'll try and work some of those into the conversation as well. Finally, you can find some answers to common technical issues located in the help widget at the bottom of your screen. And with that, I'm Jack Jacobson, and I'm an advisor at Thompson Coburn and one of the Council for Federal Cannabis Regulations advisors as well. Today, we're going to dive into the recent report findings from the FDA on quality standards in state programs permitting cannabis for medical use. Specifically, understanding the quality of cannabis found in these programs, the collected laws, regulations, and guidance documents available on public state-run websites, and how they compare with current good manufacturing practices applicable to finished drug products. We've gathered together a dynamic panel today, and now I'd like to introduce and wish a happy birthday to my co-host for this series, Sarah Chase.
Well, thank you, Jack. Good afternoon, everyone. I'm Sarah Chase, the Executive Director for the Council for Federal Cannabis Regulation, and it's my sincere pleasure to welcome you to the latest installment of CFCR's webinar series. And I encourage all of you who are watching and participating today to ask a lot of questions, and we'll try to get to as many as we possibly can. And I know that some of you pre-submitted them, and the questions that we've received have been put into today's roster for the guests. And also, but please use the Q&A widget to submit your questions if you have them during the webinar series today. And for members of the press, you can send direct questions through the email address that I'm going to try to put in the chat for you a little bit later on. We can't take direct questions from the press today, but again, we'll provide you with that email address where you can send those in directly. And we have two expert speakers joining us today from the U.S. Food and Drug Administration to help us all better understand the complexity surrounding quality standards, and especially as they relate to medical cannabis products. And Jack and I would just like to acknowledge the important work of our guests and the agency in compiling this analysis that we're going to be talking about today. And we're all here because we care about health and safety standards, and the efforts undertaken by today's guests are instrumental in providing all of us with greater guidance. So we'd like to thank the FDA for their engagement on this topic and for effectively advancing a more clear understanding of regulatory science, research standards, and how this will ultimately inform the regulation of emerging industries and products. So with that, let's take a moment for each of our guests to briefly introduce themselves, starting with our returning guest, Dr. Cassandra Taylor.
And Sarah, I think Cassie might have lost audio, so if Skylar could start, that would be great.
Oh, yep. Go ahead, Skylar.
Yeah, of course. All right. Hi, everyone. My name is Skylar Prine. I joined FDA in the Botanical Review Team in 2019. I worked very closely with Cassie, who unfortunately couldn't give her intro first, but Cassie is a cannabis subject matter expert for CEDAR and the agency. So I worked very closely with her, assisting in various cannabis initiatives within the Center for Drugs and across FDA. And then in January of this year, actually, I moved over to the Office of Executive Programs. I'll refer to that as OEP, which is still in the Center for Drugs. And OEP's primary function is to support the Office of the Center Director. And we do that in various different ways. And we facilitate information exchange among the center's internal and external customers. We coordinate hearings, briefings, meetings, correspondence, intergovernmental inquiries, etc. And we also carry out a bunch of different priority assignments and special projects for the center.
Excellent. And I think we're still trying to get Cassie's audio back up on here. But Skylar, maybe you could start by telling us a little bit about how this analysis came about, and what was the genesis and the purpose of it.
Okay, of course. So we were aware that there is an increased public interest in using cannabis and cannabis-derived products from state-regulated dispensaries in human clinical trials. So we really wanted to better understand the types of quality-related tests that these products undergo at the state level. We were also interested in understanding how the various state regulations on testing for cannabis and cannabis-derived products accessible in these state programs compared to our regulations for human drug products. So in other words, how did they compare to CGMPs? And we really wanted to put together a work that would inform those developing cannabis drugs about the differences between the state requirements, some of which they may be very familiar with, and FDA's expectations for investigational new drug applications.
Excellent. And I hear that we have Dr. Taylor back on. So Dr. Taylor, would you just take a moment to briefly introduce yourself?
Sure. Sorry about that, everyone. I'm not at my home office. I'm on vacation, not at the beach, but I apologize for that.
Oops, I think we've lost her again. Well, we're going to do some background refresh here real quick on the audio. And again, thank you to Dr. Taylor, who is taking time off of her vacation to join us today. Skylar, just to kind of pick up on one of your points there, when you're looking at the state programs, what was the process in collecting some of the data there?
Yeah, so, we actually started this process, back in late 2019, early 2020. And I did what most of us would do. I turned to the internet to find out if anyone had already done. Oh, is Cassie back? Hey, Cassie, you back on?
I'm back. I'm sorry.
Yeah. No, okay. You want to do your intro now while we have you?
Yes. Can you hear me?
Yes.
Yep. Okay, I'll be fast. I'm Cassie Taylor. I work in Cedar. I'm on the Botanical Review team. Cannabis subject matter expert. And I work across Cedar with lots of different folks. Clinical space, but also folks who do not. So I will keep it short so we can get to the questions. Sorry about the technical issues.
No worries.
Awesome.
And Skylar, if you want to pick up just on that question on state programs and what was the process on collecting the data?
Yeah, so, when I did my first, dive into this, I was unable to find a comparison across the programs, specifically for the quality-related testing. So that's when I began, collecting them myself. I spent a long time combing through state-run websites to find where they posted their laws, their regulations, their guidance documents, etc. And once I had a complete collection of documents, I began looking at each of the programs and comparing what I saw written for each of the quality-related tests. And for the publication, we decided on a drop-dead date of June 30th, 2021. So on that date, I went to each of the state-run websites that I had familiarized myself with for about a year, and downloaded all the related documents and content that was posted and active as of that date, and that's what we used to put together our publication.
Excellent. Is there any reason why you chose the drop dead date of June 30th, or just had to have a cutoff?
Yeah, so as everyone the state, what's going on in the states, is changing every day. So we had to pick. It would be unfair if we collected half of them in May and half of them in June, and then compared them. So we wanted to have one date where, as of this time, if we froze in time, this is what was going on at the states. And of course, this, when the publication came out, was a little bit after June 30th, 2021, but at least, we had a snapshot of what it looked like at that point.
Great. And I've got one more follow-up question for you, and then I'll throw it over to Jack here because I know he's got a bunch of prompts for you and questions. I think the audience is going to want to know, why did you focus on state medical programs and not just adult use or recreational use?
Yeah, so, our main goal of this publication was to provide considerations to facilitate the development of consistent, high-quality products for potential use in clinical trials of drugs. So, that being said, we focused on the medical programs since, patients who are registered in these programs are presumably using these cannabis products for a medical indication that could, potentially be studied in human clinical trials. So, starting at starting by looking at just the medical programs made the most sense, for our goal. Especially since, as I mentioned earlier, we, really wanted to highlight FDA's expectations for IND applications. CDER, the Center for Drugs, remains convinced that the best way to explore the therapeutic potential of these products is through FDA's IND pathway.
And what did you discover? What was sort of interesting about this that's going to ultimately inform IND?
So I think, as we did our comparison, I know Cassie's going to jump in and talk about, some of the comparisons we did to USP, but, when the FDA is reviewing some of these applications, there's specific areas, where we're looking at what needs to be done in order to have these quality products. So I'm going to let Cassie do some of the deep diving on all of that, but we really wanted to highlight, what these products would be analyzed for within FDA.
Great. Thanks, Skylar. And just for the audience, Cassie's still listening in, and we'll jump in when she's able, but she's not going to have video moving forward. Speaking of USP, can you explain the importance of USP methods and why you drew from them in your analysis? Skylar?
Yeah, so, for,
Skylar,
the USP...
can you hear me?
Oh, yes, I can hear you, Cassie.
Yes, I can answer that one, Jack. I apologize for the technology issue, everyone, because there's just nothing I can do about it. But in terms of why people would use USP versus something else, USP is very well established. They have a standard setting. They're an institute, have a really transparent, well-known process to set these sort of standards. And many people we appreciate our USP colleagues' work because it's not a work as they can the task to. So the laborious task of updating a lot of these standards using the established we drew on the USP methods is because USP methods, yes.
Thanks, Cassie. You're still cutting in and out a little bit, so maybe we can ask Skylar to fill in any blanks or add to.
Yeah, so, as Cassie was saying, like our USP colleagues spent a long time putting together these standards, and for the USP methods, any of them under 1,000 are binding and legally enforceable, and those over do not contain any mandatory requirements. So the ones that we use we compare to in our paper are using those that we would be looking at when reviewing drugs under an IND application.
And just so the audience knows, Dr. Taylor is going to try to dial in from her phone so we can get some better audio here going forward. That'll take just a second here. Well, Cassie, Skylar, why is it important then for labs in particular to sort of train their staff on USP methods?
Well, so if labs who are, looking at these products that are in these state programs are testing these products, and if these products are potentially going to be used in clinical trials, we would be looking for these methods to be used and for these tests to be done. So in order for your products to, be looked at by FDA, it would be really helpful to be aware of these USP standards and these USP methods.
Okay, and I think, Dr. Taylor, just to clarify, you are in on audio now, right? Can you hear us?
I am here. Yes. Are you guys able to hear me?
Yep, perfectly.
Okay, wonderful.
Jack, if you want to continue on here, I know you've got some questions.
Yeah, and thank you, Cassie, for being flexible for us. If I'm a CRO, what's the takeaway from this analysis, and how does this relate to IND submission?
Great question, Jack. I do apologize that I missed Skylar's response, but something quickly I wanted to point out about those USP methods is just a reminder to everybody that anything under the 999 is enforceable for drugs, and everything over that is non-enforceable. And so if you're a CRO, that's important to know. Why is that important, you might be asking? Okay, so if you're a CRO and you're providing testing services to researchers who maybe want to utilize the product under an IND, you might want to consider training and qualifying your staff on those USP methods, because those are the minimum method standards for drug development and research. When you come into FDA and you use a USP method, we can always ask you to do more, but the minimum standard is those USP methods. And so if you're a CRO and you have someone coming in and saying, hey, could you please help test this for me? I'm planning to send it in or use it under IND. Having those USP methods already available at your CRO is a really great base level option for those research clients that may be coming into you. And as a really quick reminder, when you have an IND and you come into the FDA, there's kind of three basic requirements that you have to meet. The first is the chemistry, manufacturing, and controls. So that includes the botanical raw material information, quantitating values of cannabinoids and terpenes and other phytochemicals. And if you have any sort of manufacturing of, let's say, a dried botanical, like a cannabis flower, and then you're maybe performing extractions or some other sort of manipulation, you also need to have those methods and that information quantitatively available when you submit it to the IND. Additionally, you have to have farm tox and clinical information, which is the discussion for today. But if you're doing research and you go to a CRO and they already have those USP methods validated and verified, it's a great place to start. And that actually helps your relationship with those researchers, because then they already know that you have taken the time in your laboratory, you've also qualified your staff, you've trained them up, and that actually helps the researcher utilize you as the CRO to provide the appropriate information that they could submit under IND. And so I have worked at a CRO myself. I know it's sometimes really challenging to take on new methodologies, but USP methods are very well established, and oftentimes they can assist you. They have lots of information there, help you with your quality control teams to get set up. And sometimes I know, as well as all of you, that methods need to be fit for purpose, and you have to sometimes make little tweaks and adjustments. But it's a lot easier if you can start from something that's really well established, like a USP method, and then all you have to do mainly is mostly verify those small tweaks. So maybe you're changing the solvent system slightly. Maybe you're changing a runtime. Maybe you have to change some sort of like sample preparation. That's okay. All you have to do is denote that you do it. You have to do your normal QA, QC methodologies from your QA managers, and you can verify those methodologies. But you're starting from something that's already established, and if everyone is using that same methodology, it's a lot easier to compare and contrast, especially if your researcher is really looking for you to have a specific type of information.
Great. And Cassie, a follow-up question to that too, that's coming in from the audience is what's the difference between USP methods and standards versus AOAC or ASTM?
So for CEDAR, the difference is that USP methods that are under 1,000 are actually enforceable methods. So like I had stated for INDs, those are requirements. That's the minimum requirement. Whereas ASTM, AOAC, also wonderful standard setting organizations, but they don't have the same enforceability from the FDA standpoint, because the USP methods are the ones that are drug monographs that are under 1,000. Those are the ones that are enforceable. You can use those other methodologies. That's okay. There's nothing wrong with that, but you need to make sure that you're doing proper method validation and making sure that you also are meeting the minimum requirements. So if, let's say you're trying to use an AOAC method as an example, instead of a USP method, you're going to have to demonstrate that it is the same or better when you come into FDA. So that's something you have to keep in mind. But that's why we recommend starting with the USP methods, because that's the bare minimum, and those are enforceable on the drug side of things. Does that make sense, Sarah?
Yes, it does. Thank you. And I guess that's a lead-up question for me too. Is why is the original monograph filed on cannabis at USP no longer valid?
Great question. So that monograph, I actually had to look it up. It's from 1850, and it was actually a recognized drug monograph in the U.S. Pharmacopoeia. And so it's always been a drug monograph, and it hasn't been anything other than a drug monograph. Back in the 1850s, obviously things were very, very different back then. The cannabis was definitely very different, and it wasn't necessarily even remotely close to the cannabis, I think, that we're seeing today. But there really has not been like a cannabis monograph published since the USP, I think, omitted that one in the early 1940s, in mainly a response to a lot of the laws that were being set forth in the late 30s. I think it was 37 was when they first started establishing a lot of those narcotic drug laws. I'm not a lawyer, so don't quote me on that exact date, but it's somewhere around there. So that one from the 1850s really was always a drug monograph, but it's really not it's not a fair comparison to what we're really seeing today. And really, I think the way that we regulated medicine is extremely different than there really weren't a lot of regulations back then in the 1850s. So it's something to just kind of keep in the back of your mind. But USP is obviously better to discuss that. I know Dr. Nandu Sarma has talked about this in past open forums and other places, that this was an established drug monograph back then, and it is no longer available in the pharmacopoeia.
Sarah, I think we've got one more related question. Have USP method authors validated USP methods for cannabis matrices?
Have USP authors, say that again, Jack, have USP authors validated what?
This is an audience question. Have USP method authors validated USP methods for cannabis matrices? I
don't know, because I'm not part of USP, and I should clarify FDA's relationship with USP. So I will answer that, because I can't answer this question, but USP has their expert committees, and FDA serves as a non-voting member. So for those who have participated in expert committees, I know that there is one on cannabis, and they have brought a lot of the experts in the field into that committee. FDA is there as an observer, and we will provide FDA input on what our current thinking are, just like we do for guidance, to ensure that whatever they're creating is in alignment with FDA's current viewpoints, especially if something is going to be enforceable. So in a monograph, that would be under that 999. So that's something to think about. So we don't actually vote there. And so we are not actually in on all of those discussions, because as regulators, that would be a conflict of interest for us to participate there, because we actually enforce those monographs under our drug laws. And so we really cannot be involved in making those decisions, which is why USP as an organization is completely neutral party. And FDA is just there to say, hey, you guys have done your work. We'll review and make sure it's in alignment with our current thinking on whatever the process might be. It could be elemental impurities. It could be pesticides. It could be any number of things in the drug space. And so FDA will provide their opinion based on what the committee creates themselves. And so that would be a great question for Nandu Sarma, who can answer that, because FDA really just can't be involved at that level.
So, Cassie, then how is implementation sometimes differing, or why is it different state by state?
Yeah, that's a great question, Sarah. So what we did, and I think Skylar had talked about this earlier, is we went and actually looked at the regulations that are on the internet in different states. And we obviously notice that they're different from state to state. But what appears is that different states have different resources that are available to them. They have different personnel that are available to them. They have different analytical instrumentation that is available to them. As the list goes on and on and on, for everyone who works in a laboratory understands these challenges. And so what seems to be is that the implementation didn't always necessarily match what was there in the regs, and that's no one's fault at the state level. It's not the laboratory's fault. It's just sometimes it appears that it was just harder to actually implement those things. And I think a lot of that was because of resources, which I think everyone on this call understands resources are a challenge, not just in laboratories, but across, federal, state, local, tribal territories. This is a very challenging space, especially given the fact that we've been in a pandemic for 2.5 years. Resources are getting pulled into other places. And so sometimes implementing some of those regulations, not that they're not doing it, but sometimes it's delayed for a variety of reasons, because these laboratories are not the people that are legislating what is written into their state laws. They're the scientists who are then trying to implement whatever is written. And so, as I think we've all seen, Sarah, I know your team has reported on, New Jersey signs into law, but then it still takes time to really implement the system. It's the same thing when there's new testing standards. It takes time to implement those, just like if you find out as a CRO, oh, now we have to start doing a pesticide testing suite. Well, you have to get everybody trained up. You have to get all the instrumentation qualified. You have to get things verified. That takes time. And so I think that's likely one of the reasons why sometimes the implementation maybe lags a little bit behind what is in the regulations that we're finding online.
Well, then maybe both of you could build on this a little bit too. So if you're testing for a medical state program, what should you really be considering then, based upon what you found in the report and the analysis?
Skylar, do you want to highlight the test that we talked about?
Yeah, so, if you're, in a medical program and, like we said for this paper, we were really trying to highlight what we're looking at, what FDA would be looking at for products coming into us through an IND application. But if you're, planning to use a product that is found in these state markets under an IND, you will want to use some of these tests, like looking at heavy metals, looking at pesticides, looking at microbials, and really, really think about, what's written into USP and using those types of methods, looking at the types of analytes and using those when you're testing your products before submitting them. And Cassie, if you want to add on to that too.
Yeah, I think there is a lot that people, are testing for, and then there's things people are not testing for. And so we don't dictate what happens at any of the state level. And so it's challenging for us, I think, to say what people should or should not do. We try to look at it from, if you were to come in under an IND, what types of information, would you need to submit? So definitely, we want to know what your cannabinoid profile is. If there's other phytochemicals that are present, terpenes, maybe there's other fatty acids, things that are available, making sure, and anyone who's had experience in the IND space, our CMC colleagues often talk about making sure that you have mass balance. And so making sure that you account for as many of the compounds as you can. That's reasonable. We're not trying to give anyone an undue burden, but having an understanding of those types of things from a botanical perspective, in our guidance, we talk about good agricultural and collection practices. And so making sure that you are testing for pesticides, aflatoxins, adventitious agents like, microbes and things of that nature are all really important, because we need to make sure that whoever participates under an IND in a clinical trial, that those people are safe and that they won't have to deal with any sort of adventitious agents that are entering their bodies while in a space.
Thank you, Dr. Taylor. And another audience question regards FDA's position with natural variation in active plant ingredients used in botanical drugs. In reading the guidance, FDA understands this phenomenon, yet there is no specific variation given, e.g. 10%. So it appears on a case-by-case basis, although, quote, it may be permissible in unusual cases to augment levels in a botanical drug to achieve batch-to-batch consistency. Further, it appears FDA recommends each issue be separately addressed in consultation with the FDA. Can you speak to this in general or in cannabinoids in particular?
Thanks, Jack. Yeah, I can talk about that. So the guidance does say that, and really the best way for you to get specific information on either a cannabis product or any other botanical product is definitely to come to the FDA. And one of the best pathways that you can utilize is called the Pre-Ind Meeting. So as a general reminder, when you come to FDA for an IND, once that IND gets stamped inside FDA means you checked all the boxes, the clock starts ticking. It's 30 days for a decision. So either you will get what's called a safe to proceed, or you would put on a clinical hold or a partial clinical hold for a variety of safety reasons that can come from any part of the review team. The good part about a pre-IND is that there is no clock and that you can come to the FDA and let's say you want to study a cannabis-derived product for some oncology indication. You would write to our oncology division, which is in the Office of New Drugs, and you would say, here is my planned protocol, here's where we're at, here's the type of cannabis product or products I would like to use, and here is my questions. And you ask your questions, and then the division reviews that request. They either will grant you what's called a written response only, or they will grant you a TCON. And then we can kind of talk through a lot of those questions. And the reason that we do it that way is because each one of those protocols is very specific. You have to take things into consideration, like who is going to be dosed in your clinical trial? Are they healthy people? Are they people who are sick? Are these people in the cannabis space, have they ever consumed cannabis before? Are these naive users? Are these users who have used cannabis for quite some time? What type of product are you going to be dosing people with? Have people had access to this product in the past? And so these are all things that we have to consider, as well as pharmacology, toxicology information to assess the safety of any botanical. And so using the pre-IND pathway is the best way for you to get very specific feedback on the exact protocol that you're proposing to do.
Thanks.
And it looks like there's a follow-on, Jack.
Yeah, there is. I think this fits in here. Do the scientists believe that the current regulatory framework for pharmaceuticals is appropriate or otherwise sufficient for approval of non-isolated botanical extracts and whole cannabis flower?
So yes, we feel that the botanical drug approval pathway is appropriate. There are 2 botanical drugs that are already approved. They are not cannabis. It's Verogen, which is our green tea, and crofelemer. And so it is possible for you to come to the FDA under an IND, work through phases 1 through 3, through drug development, talking to FDA through the botanical review team, as well as our CMC colleagues, our farm tox colleagues, our clinical colleagues. And then we can work through those things and those challenges with you that do come up, because no one here is pretending that it's easy to do botanical drug development. It's not. It's challenging, and it's not for the weak of heart. Anyone who's worked in natural products knows that natural products are very challenging to take on. But we do really believe that the best way for folks to look at these botanicals is to utilize the IND pathway, and there is a drug pathway for it. We just understand that in your beginning phases of drug development, there are different things that have to be taken into account, and we do understand that. We do work together with you, which is why if you use that pre-IND pathway, you can get very specific feedback on what you personally would need to do for your own drug development. Even if you're not intending to, submit an NDA, maybe you're just doing research, you're still going to get that specific feedback from us to help you get to a place where you can hopefully proceed safely and to start whatever clinical trials that you're proposing for us.
Cassie and Skylar, Delta 8, Delta 9 is very much on everyone's mind right now. Maybe could you address some of the issues you're seeing around Delta 8 quality standards testing and or with other cannabinoids other than Delta 9?
Sure, I can talk about that. I think, Sarah, just for everyone, I think Sarah's implying this, but just for those who don't know, we've obviously put out some warning letters recently for Delta 8, which I think everyone saw. It was about 3 weeks ago. And we recently put out some information about some THC edible products that children have been ingesting, those sorts of things. And so we are acutely aware that these products are out on the marketplace. In terms of testing, I do think, like we talked about at the beginning, obviously there is not a lot of standardized testing really for Delta 8 because it's quote unquote new, right? It's kind of been emerging into the marketplace. And so I do think sometimes that it's challenging, and for people who perhaps don't have as much experience in natural products may not necessarily understand how challenging it could be to be testing Delta 8, or also may not truly understand that when Delta 8 is being synthesized from CBD, that lots of other compounds are actually being created as impurities and as byproducts. And for those who are not chemists, that transformation of the molecule of CBD to Delta 8 THC has to occur under chemical synthesis. It's not naturally occurring in any large amount. In the plant itself, it's very, very small, like almost trace amounts, like less than 0.1%. It's quite low. And so when folks are creating these types of compounds, they really, I think, don't necessarily always understand that they're also creating more than just Delta 8. And if you don't test for something, you're not going to find something. So if you're only putting in your HPLC a Delta 8 and a Delta 9 standard, and you're not putting in anything else, you're not going to see anything else because you don't have those standards. And I'm not suggesting that folks are doing that, but if you're not looking for these other cannabinoids or other products that are being formed, you're not going to find them. And so it's something to keep in mind as labs are starting to create methodologies and quantification of Delta 8, Delta 9, other tetrahydrocannabinols that maybe they're seeing in products that are coming in, which is why I think it's become challenging because there really is no exact method that folks are using. But I do know other standard setting organizations are working hard to get some methods available to the public so that everyone could start at least on the same level and then make them fit for purpose.
And we have a related question sort of on the science side of things. Similar to prescriptions, if physicians were able to control the exact amount of every ingredient in a medical cannabis formulation with pharma-grade precision, and even titrate the amount of THC per patient in 1 milligram increments based on the patient's feedback, would the FDA be more inclined to approve medical cannabis?
Well, again, you would have to come in under an IND for me to actually know what you're doing. I know that sounded very specific, Jack, but there's an excessive amount of information that is missing from that statement. And I'm not disparaging at all to the person who wrote it. I really do appreciate that question, but I think it goes to the point of what we were just talking about is it's our duty here at the FDA to make sure that when you come in and you're giving other people something that you're intending to diagnose, treat, cure, mitigate, or prevent some sort of disease, we do have to know everything about it. And so, yes, that point is very important. Controlling those different cannabinoids is super important. And it's important in botanicals, which we've always talked about. That's why we really work hard with sponsors to help them establish those good agricultural and collection practices, help reduce those variables as much as they can. And then if researchers want to take it a step further and take their botanical dried material and maybe do some extraction to maybe get rid of a couple compounds that they personally are like, we don't want to study these, that's fine. We work with you through that. Our CMC colleagues are wonderful. They can help you through that, get to, making sure your drug substance acceptance criteria is reasonable, making sure you're using reasonable methodologies. And then in your final formulation step, we have drug product formulation specialists in CMC that can assist you to make sure that you're getting there. And so it's, we want people to feel that this is an approach that has been successful, and we are here to work with you. We're not trying to give undue burden to folks, and we do believe that these products can be approved as long as you're meeting those IND requirements. And then if you so choose to go on to the NDA steps for a marketing drug application, there's a pathway for you to do that. And so you're not taking this journey alone. You're coming to us and we're providing you with the information that you need to get to a place where you can dose patients safely. And as you progress through those phases of drug development, that's when things get challenging. You need to start figuring out how you're going to demonstrate efficacy. And demonstration of efficacy is a challenge for not just botanical drugs, but even single molecule drugs that are a compound just by itself. And so that's why we start with safety, and we work together, to help get information that is necessary to meet those requirements.
Mm-hmm. We're getting quite a few audience questions just about some of the links and references that you mentioned in this, and we're going to, in a few seconds, just drop the link to the analysis report into the chat for everybody, and that's gotten most of the links that both Skylar and Cassie have referenced today, too. And quickly, we've got an audience question. I want to move back just as a point of clarification when we were talking about USP and ASTM and AOAC. Audience members got a question to clarify, just to make sure FDA can mandate ASTM methods in certain circumstances. Is that correct?
ASTM methods are not enforceable. USP drugs, monographs are enforceable. However, if you come to us and you want to use something that's not a USP method, you have to validate that method. You have to share the method validation with us, and then we review it and tell you if it's acceptable. That's all part of the IND process. Our CMC colleagues do that all the time for botanical and non-botanical drugs.
Thank you for clarifying that. And then one more question, and this may not be one that you can answer, but it's an interesting one. Will the FDA, or in your opinion, will the FDA ever consider a smokable flower product for IND approval?
Well, we can't really answer any questions about what's going to happen in the future. All of those decisions and regulatory decisions are made through official regulatory pathways. So we can't answer that unless you come in under an IND and we can talk you through the process.
Okay, thank you. So when we're talking about public health protection, how are these quality standards impacting the surveillance of public health protection?
Repeat that again, please, Sarah.
How will these quality standards impact surveillance and public health protection?
So the quality health standards like that we talked about, those are at the state level. We don't have jurisdiction at the state level. And so a lot of our surveillance is based on approved drug products, but we also have, the systems affairs and cares, which are adverse events. And so people can report adverse events for unapproved products into those systems. But I think each state would have to speak for itself because they have very different systems, right? They have laboratory systems, they have departments of health, and they have different capacities for enforcement and surveillance. And some programs have a large budget for that, some programs don't. And so I think that's a challenge for our state level colleagues. I think that's the most we can say about that.
Is there one particular state that you've observed that is sort of very well done, that would serve as a model?
We're not going to pick favorites, Sarah.
No, we're not going to pick favorites.
No, I think
there was just one state that actually had some sort of laws that remind me. My mind is blanking.
Yeah, no, I mean, through this analysis, we really found out how different, right, it is across the different states. And I don't think that's to say, one is better than the other, one's the best, one's the worst. I think that, this process of learning really what's actually going on is what's important. Like understanding and, understanding what the differences are, up front, and then, figuring out, right, where maybe in the middle are we supposed to be for these type of tests.
Jack, did you have a follow-up to that one?
Yeah, I was just going to follow up specifically on that to get back a little more into the report side rather than the FDA side, the FDA process side. What specific actions can the cannabis industry and state regulators and policymakers use the information in this paper to better protect consumers in ensuring the safe and consistent production of products? And if, Skylar, you'd like to start, and then Dr. Taylor can chime in after.
Yeah. So I think, as kind of going off of what I was saying with the last question, it's just trying to get the consistency among these products and thinking about, what drug products that come into FDA, the assessment that they go through, and what FDA looks at for any other drug product, and thinking about that in this context for these products where there are registered patients who are in medical programs who are using it for, whatever indication, these products, you would hope and you would want them to be having or going through the same assessment that any other drug product that you would be taking goes through. So that was really, what we hope. And, I hope everyone, would feel that they are taking a product and that it's at the highest quality. And then when you take it, in the morning, it's the same as when you take it later in the day. So that was really the goal here.
Yeah, and I would echo Skylar that I think it's really important if it's feasible at the state level for folks to start doing their best to use the same sort of methodologies to try to ensure that consistency. So it's not just about safety, but it's also helping to set those quality standards. And so folks can utilize these specific methods to get them started to set up their various quality systems. And hopefully that will allow the different systems to work together. So that way folks feel like if they go from one state to another state, that they're getting the same sort of product at the same sort of level that maybe they have gotten in their home state.
So how then does the FDA plan to evaluate the varying kind of multiple active ingredients of botanical cannabis products?
Yeah, I can answer that, Sarah. So we already do that. It's under the same framework that we've talked about for other botanicals. So I think everyone knows botanicals are very heterogeneous mixtures. They can sometimes have, thousands of compounds. We're not asking anybody to check the 1,000 compounds, so don't worry. But how we look at that is we take a totality of the evidence approach with every botanical, not just cannabis. And we do ask you as the researcher to really focus on the compounds that you believe to be active in whatever product that you're proposing for drug development or for research. And we ask you to quantitatively assess those. And then that information comes in during the IND process. CRT as well as our CNC colleagues, we review that information. Maybe we have more information requests that we need to ask you about to make sure that you're meeting the minimum safety requirements. But in general, we look at all of those compounds holistically. And so here at the FDA, for any botanical that comes in the door, the entire botanical drug is considered active. And so everything that's in there is considered active. And so if you are not able to identify some of the information, we can talk with you about that. There's different things that you can do, things like the biological assay. If you're trying to figure out, well, we think this might be active, but we're not sure. We can work together with you on that to help you get to something that makes sense, because sometimes it's just really hard to quantify specific compounds. It's just sometimes really challenging, and we recognize that. And so that totality of the evidence approach is not unique to cannabis. It is unique to botanicals, but we just understand that there's a lot of challenges in that space, and we do try to work together with folks.
And with the totality of evidence approach, would that also to apply to veterinary products or animal-based products?
No, that's for human drug products only, Sarah. So veterinary products, they have a different set of standards, and so that goes for our Center for Veterinary Medicine. That's a totally different application type. They have both veterinary foods and veterinary drug applications. I am not an expert. That's literally the limit of my understanding. But just like dietary supplements and foods, that's a different center. They have a different application process. They have a different standards process. So that totality of the evidence is for botanical human drugs. Thank you for clarifying.
Yeah, that makes me think one of our next webinars might be with the Center of Veterinary Medicine over there too, because the warning letters came out today about CBD and animal products. So I think this is a top of mind question too. Jack, did you have a follow-up?
I literally know nothing about that product, so I apologize. I can't help you there.
No. It's good clarification. Thank you. And we should have pointed out too earlier sort of the differences between cedar and sifsan too for some of the audience members who may be a little bit new to this. I don't know if you want to delve into that quickly.
Oh, sure. I can do that really quick. So Skylar and I, we work in CEDAR's Center for Drug Evaluation and Research. We're the largest center in the FDA. And so we regulate human drug products. So that includes prescription drug products, generic drug products, and over-the-counter drug products. One of our sister centers is the Center for Food Safety and Applied Nutrition, or CFCR, and they actually regulate food, additives, dietary supplements, and cosmetics. And so we have different regulations in CEDAR than our colleagues in CFCR, as well as our colleagues in CVM, who we just talked about, Center for Veterinary Medicine. There are different regulations for the different product categories. And so drugs generally is the most stringent. And so here in CEDAR, that's why we focus really on the human drug products, as that is our charge and what we have jurisdiction over, where our CFCR colleagues have jurisdiction over a wide variety of products, and they have to meet a different set of regulations. Super high level, Sarah. Hopefully that's helpful to folks.
Thank you.
And we're getting really close to the hour, so have one or two final questions. Would love to know, this is sort of a groundbreaking report by FDA, so what are some of the general feedback that you've been receiving about your analysis, and what's next?
I will tell you, Jack, we were pleasantly surprised that it was positive feedback, because being at the FDA, we're pretty used to negative feedback. We mean that in the kindest way possible. It has been really positive feedback, and I won't speak for Skylar. I'll give her a minute to talk about it, but I felt really good about the fact that we kind of are starting to fill these holes of the science, and if we can at least do our part doing an analysis like this, we would love for others to jump in and to help continue the work moving forward. But I want to give Skylar a moment to talk about it, since she's our lead author.
Yeah, and I say like one of the biggest things too is that we welcome the feedback. We do these things based on what, the questions that we have, right? But I think the more feedback we get, the better we can put together, more topics, more publications like this, and feed off of this starting point, because this is just the starting point, so we really do welcome the feedback, and we appreciate, anyone who has already provided us feedback, but we'll take it, the good and the bad. I think that'll help us move forward.
Sarah, do you want to take those clarifying questions, and then we'll wrap up, I think.
Yeah, we've got a few more in here. And just for everybody to know, I am dropping the link for the PubMed publication in here for you. And also, we'll state this later, but the whole entire video will be available on our website later on, too, for everybody to download off of YouTube. see, we got some audience questions coming in. Can you ever anticipate, this may not be one you can answer, but would you ever anticipate vaping systems being used to deliver medicines?
Yeah, Sarah, thanks for that question. So there are inhalational routes of administration that are approved for all different types of drug products. Think of things like an albuterol inhaler, if anyone has seen anyone with asthma use that. So if someone were to consider using a vaping device with a drug product here at the FDA, that's called a combination product. And so that would involve not only CDER, but our Center for Devices and Radiological Health. They would help assess any sort of device like that, whether it be a vape or something else. And they would then provide you the specific regulations that are needed to approve that device in conjunction with the drug, that information would be provided by CDER. So those pathways are available. We just would have to see what people come to us with. That's a good question.
Great. Okay. One other follow-up question from previous audience submissions here. Well, what current markets and their CGMPs can be compared to that of the developing cannabis market?
Like what other product markets?
What current markets and their CGMPs can be compared to that of the developing cannabis market? So let me think of a
well, CGMPs for drugs are for drugs, and then there's CGMPs for supplements, and then there's CGMPs for food. So FDA creates CGMPs based on intended use of your product and the category in which you're using it. And so I want to clarify that if anyone comes to FDA and they want to study a proposed drug product, and let's say that they bought that drug product at GNC, let's say it's turmeric, your intended use is to come in here and treat a clinical indication. So you're now a drug. So you have to meet drug CGMPs. That's how it works. It's based on your intended use. So I don't think that we can really compare to other places.
Yep. No, that clarifies that absolutely perfectly. Thank you. And just a quick reminder, audience, we're coming up to the last couple of minutes here. So if you have any more questions, please let us know. Otherwise, Jack, I'll hand it over to you.
Thanks, Sarah. Skylar, any last observations or anything that we didn't get to that you'd like to share with today's audience?
No, I think we had a great conversation here today. I think there was a lot of great questions. And, just reiterating what I said before, we appreciate the feedback, we appreciate the questions, and, feel free to reach out to Cassie and I with any questions or feedback that you may have. And, we really appreciate people joining today and participating in this kind of conversation, and for, taking the time to look at our work. It's, very exciting to have these opportunities.
Thanks, Skylar. Cassie, any final takeaways for you?
Yeah, thanks, Jack. I also want to extend a thanks to everyone who's taken the time out today. I also want to apologize for my technical difficulties, and sorry you can't see my face, but I really have appreciated the feedback from the community. The scientific community in the cannabis space has really provided excellent feedback to us, and we really do take it seriously. So when Skylar said reach out to us, you can definitely reach out to us. Please let us know if you have additional feedback. We know that this is just the start of the work, and we hope that the rest of you who are in this space are looking to help carry the work forward, because we know that there's a lot of work to do. So this is just the beginning. But thank you so much for everyone who really took the time to read this article and really try to do your best to create these questions for us. So thank you for the opportunity. I also want to thank Sarah and Jack for providing this opportunity. That's been one of the positive things about publishing this paper, is we get opportunities like this to talk with great people and also to interact with folks on the phone. So thank you, everyone. I know we didn't get to all the questions, but I really do appreciate you engaging with us on this topic. So thanks so much, Jack.
Thank you, Dr. Taylor, and thank you all in the audience for joining us today. Our next webinar will be held on Thursday, June 30th at 1 p.m. Eastern, and we'll be joined by 3 leading experts in lab testing, so very timely, and they'll be sharing their perspectives on the unsettling rise in testing fraud within the industry. Sarah?
Well, I can't thank you all enough for joining us today, and especially you, Cassie, for joining us on vacation and making the time for everyone today. And Skylar, it's been a pleasure getting to know you, and we hope that you'll be back many more times too, because this is important work that you're doing, and it's great to highlight all of this, both for our audience and for the industry. So thank you all again, and for being great communicators about good science. And as Jack mentioned, our next webinar is all about lab testing fraud, and this is definitely going to be a very engaging conversation. And you can go to www.uscfcr.org/events for more information. And pre-registration will open in just about a week or so. We'll send out a little bit of a news announcement about that. And also, as I mentioned earlier, a video copy of today's webinar will be posted on our website and on our YouTube channel. It'll probably go up early next week. It just takes us a few days to download everything and get it ready. So you can share it with anybody who was unable to attend, or if you want to go back and get some notes from it, please feel free to listen to that webinar over and over again. And again, it will be up on our website at www.uscfcr.org, and you can find that on the YouTube link in the social media links there. So, and Jack, I'll throw it over to you for a final word. And again, thanks to our guests.
And thank you everyone for making today's a big success. And we'll see you next month. And if you're still with us, can you please take a moment to complete our survey at the end of this webinar? Thank you. Thanks, Skylar.
Take care.
Thanks, Cassie.